Are my slides visible?
Yes,
Okay,
perfect.
great. Awesome. Well, thank you, Orly, Florian, and Isidro for organizing this and actually inviting me for speaker. It's big honor and really exciting lineup in the next coming year. So great. So I want to illustrate a concept in this talk. But then also show you some places where we've applied it and hopefully convince you that it's a useful concept to sort of adopt as a standard approach to understanding genomic rearrangements, meaning structural variation, copy number alterations, but also thinking more broadly about genome structure, particularly how cancer genomes fold. Now, I almost got laughed out of the Biology of Genomes conference recently for putting this sort of overused slide. But there's actually a reason why you may have not seen it for a while, and there's a reason to cheer why you may be seeing it again. So sequencing costs were actually pretty flat for almost 10 years until 2022. And for reasons I'm not going to go into related to monopolies and the market and things I'm maybe not qualified to talk about, the price of sequencing dropped fivefold in 2023. And this is a completely constructed plot, but there are bombastic people who think that it can drop another 10 or 20 fold. And I think that kind of quantitative drop is sort of revolutionary because it opens up certain things that previously would be sort of boutique and exotic and out of our reach, and they can become routine, such as thinking about whole genome sequencing as something as routine as a CBC or complete blood count. And whole genome sequencing is actually one of the simplest assays we can do in the oncology lab. And I spend a week a month signing out clinical cases. And the state of the art these days, at least in the United States, is these targeted panels that are less than 1/10 of 1% of the whole genome. But obviously, the big challenge in applying whole genomes clinically routinely is really how to analyze and interpret the data. The data are large, about a TB per patient, probably need something on the order of 1 to 2 billion reads per case. And then really the interpretation is what's tricky. But there's a pot of gold at the end of the rainbow, which is that we can have a single consolidated test that's going to give us everything from cytogenetics to drivers to mutational signatures to zygosity and many signals that are really out of reach of the current sort of molecular oncology paradigm. And really where we've been focused is on structural variation, which means genomic rearrangements and copy number alterations. And really, this is like one of the blind spots of panel sequencing. There's actually very few copy number based biomarkers outside of HER2 or 1p19q that are in routine clinical use. And one reason for that is panels are actually, it's like a dirty little secret that panels are really terrible at copy number. And in fact, microarrays, which is a 2-decade-old technology, run circles around clinical panels. Now, whole genomes are the ideal assay. And I would say actually short read whole genomes are the ideal assay for looking at cancer genome structural variation, which might be a bit of a controversial statement. But even there, the problem is still a bit tricky. And one reason is that there are two sides of structural variation. They're actually basically both parts of a single genome structure, which is basically that genomic rearrangements and copy number alterations are very biologically codependent events, meaning that when a copy number alteration happens, it usually has some kind of rearrangement driving it, and vice versa. Most rearrangements actually produce a copy number change, whether tiny or large. And that's not just a sort of poetic, fanciful concept. It's actually a very quantitative principle that in the end comes down to physics or basic sort of chemistry or mass balance, basically, which can be summed up as this statement, which is that every copy of every segment in the genome has a left and a right neighbor, or it is at a chromosome end. And really, this is this mass balance principle. And you can sort of see it play out here, where I'm showing you 3 loci that are not adjacent in the reference genome, but have been made adjacent through rearrangement. And that neighbor, what defines a neighbor that neighbor can be in the reference, or it could be a neighbor that's distant in the reference, but actually proximal in the cancer genome. And sort of bookkeeping this is how we can achieve a higher precision of inference in inferring genome structure, but actually also extract more clearly biological signals. And really, when you go alongside this reference, this plot and reference coordinates, you see a drop from 5 to 3 copies. Well, 3 of those copies are connected to the neighbor in the reference. Where are those 2 other copies connected? Well, through this junction that connects this tiny island where we see an equivalent copy gain. And so it's not just the location and orientation of these rearrangements, but actually the delta of copy number that is enforced by this mass balance signal. And we've implemented this in an algorithm that really works genome-wide by solving an optimization problem. And a key to this optimization problem is a concept called loose ends, which is really, one way mathematically to think about it is as a regularization parameter on our segmentation. But really what we're doing here is we're trying to fit the copy number data while maintaining this mass balance constraint, which really just constrains the copy number at every segment to be equal to the number of copies of incoming edges. But there are certain edges that we might miss, rearrangements that occur in repetitive regions, and we want to be robust to that. And also, we want to kind of have our segmentation minimize the number of these unaccounted for copy number alterations, or more generally, just assignments of dosage to rearrangement. So what we're doing here is we're fitting a graph which has segments and junctions, and we're assigning copy number not just to the segments, but also to the edges, while minimizing the number of loose ends. And this algorithm we originally published, actually 3 PhD theses went into this paper, and it's available on GitHub, and we've recently put together containerized sort of Nextflow based pipeline. More recently, Zining Chu, an MD PhD student in the lab, really gave this algorithm a massive upgrade, and there's a few details to this that I encourage you to look at our paper. But really, we see a step change in improvement in our ability to detect break-ends. And I think that's really important to understand how well short reads are actually, how good short reads are at this problem of detecting break-ends in cancer genomes. So here we achieve a very high precision and recall, and specifically looking at recall here, even relative to our previous Java version from 2020. And we're really ahead of the pack of these other state-of-the-art algorithms. And what really drives that is that all previous algorithms only look at copy number sort of independently of rearrangements and use this kind of change point approach. In fact, many of them rely on a tool called CBS, which we also use as input like everybody else. But these graph constraints, this mass balance, is what's really giving us additional accuracy. And you can also see this not just in the location of the break-ends, but in our ability to estimate copy number, clonal copy number genome-wide. And our recent innovations actually allow us to apply this mass balance principle in this sort of regularized context on allelic copy number, meaning parental haplotypes, which gives us additional insight into phasing. And again, we're doing really well relative to these other change point based methods. So that's algorithm performance. Now, what do we get in terms of biology? Well, one important feature that I think has been overlooked, not just in 1D structure, but in 3D structure of the genome, is something called junction copy number. And again, it's something that we're really inferring from looking at the deltas of these copy changes and inferring it genome-wide. Now, of course, it sort of might be, you may not think of junctions as having copy number, but of course, when we look under the microscope and we fish these, we can certainly see that. And one of the sort of interesting features of junction copy number is that you can have obviously low junction copy number in a region that's not very amplified. You can also have low junction copy number in a region that's highly amplified, and you can have high junction copy number in a region that's very amplified. And I think these are important biological signals because when a rearrangement is amplified, it actually means, like with any other variant that's amplified, that some other variant subsequently followed it. And in this case, these are all structural variants. So I think it gives us unique insight into mutational processes in cancer. And that's what we've used to kind of analyze the topology of not just the segments, but the junctions in these graphs to come up with, I think, cleaner criteria for inferring these catastrophic and large-scale phenomena like chromothripsis, which is a chromosome shattering, which can happen in micronuclei or anaphase bridges, and sort of reconstitution in a random order and orientation. Chromoplexy, which are these multi-way balanced rearrangements seen in prostate cancer, and TI templated insertion chains, which are copy-paste events that have been noted in multiple cancers. And these are the sort of exotic patterns of complex variation that we see in real human cancer genomes. And of course, there's been a lot of great work stemming from these observations, working backwards to mechanisms. And I think there's still a lot left to discover. Now, when we think about complex structural variants, we also may want to think about other ways of profiling these genomes, not just using these short reads that are under 1 kb. So when we talk about long reads, we're really talking about reads that are generally in the 10 to 100 kb regime. Now, ONT and other companies have come up with ultra-long reads that are longer than 100 kb, but the cost here is an order of magnitude higher for those. So it doesn't yet really scale to the whole genome in the same way that the most recent ONT sort of, the cost of long read sequencing have also gone down in addition to short reads. One of the useful tools for that next order of magnitude of molecule length is optical genome mapping, which isn't really sequencing, but taking pictures of fluorescently labeled DNA molecules. Now, longer molecules are useful for many reasons, and the intuition in the field, especially to cancer, is that we need long reads to detect most large variants, particularly these structural chromosomal alterations. That has a sort of rhyme to it: long reads, large variants, and that we miss most complex cancer structural variants using short read sequencing. And a lot of these undetected structural variants are due to recombination between homologous sequences in the genome, which we, of course, can't detect with short reads. And 54% of the genome is repeats. So really, we're missing a lot of sensitivity in hard to map regions. And finally, we need long reads to stitch together nearby rearrangements into long-range somatic haplotypes. Now, we've come into this field sort of eyes wide open. I just want to cut to the chase a little bit and plug our paper from last year, which really invalidates 3 of these 4 points. And I don't think that's bad news for long reads. I think it just helps us orient and understand a little bit better what can we expect to learn, especially in cancer, through long read profiling. And how did we get to this conclusion? One way is actually just through short read analysis. And like I told you, one reason that our method works so well is because we allow for these loose ends. We're a bit flexible in sort of allowing there to be unaccounted for mass in this graph. And these local violations of mass balance or loose ends can occur at copy changes where there's not a rearrangement, or also in these other more complex situations where we have reciprocal events and we're missing the other cognate reciprocal junction. But as you can see, most of our graph is accounted for with these junctions that we can map, and loose ends actually comprise a minority of rearrangements. Now, many of them, when we dig under sort of go back to the read data, we can find a junction that maybe fell under the filter of the initial junction caller, in our case, SVABA, or sometimes these map to repeats or novel sequences, telomeres, viruses, or repeats in the genome. Now, the sort of conjecture that cancer genomes engage in a lot of sort of repeat-driven rearrangements is based on this notion of non-allelic homologous recombination, which is that there are distinct homologous sequences that have been created through evolution, through segmental duplication, and then these can undergo some kind of holiday junction formation and crossover or gene conversion and introduce a rearrangement that will be very hard to map with short reads. Now, because of that, we would expect to see loose ends. One sort of footprint of that is that we would see loose ends where there are at sites of repeats, and indeed we see a lot of repeats around loose ends. But if there was a lot of non-allelic homologous recombination in these genomes, we would also see another thing. We would see pairs of loose ends that would occur at or near these kind of mutually homologous sequences, and we would be able to take pairs of these loose ends and use them to sort of we would see an enrichment of pairs of loose ends with homologous sequences. And in our simulation, this kind of homologous loose end pairs was a good proxy for the true number of NAHR break ends. And when we analyzed our real genomes, and our genome graphs have both germline and somatic structural variation, while we actually rarely saw pairs of these loose ends in homologous loose ends in our somatic break ends, but we actually saw them quite frequently in the germline. And that's consistent with the intuition that non-allelic homologous recombination is actually a big engine for genomic diversity across species and populations, but actually suggested that these were pretty rare in somatic variants. And we did this analysis pan-cancer. To validate this, we did linked read and long read sequencing in breast cancer and melanomas, triple negative breast cancers and melanomas, which are some of the most rearranged cancer types. And there we actually found came to a very similar conclusion, which is that we saw very few long read junctions that were spanning homologous, sort of non-allelic homologous recombination junctions among our somatic rearrangements. So here we were doing both tumor and normal, very deep sequencing with ONT and short reads. And again, we saw this signature of loose ends in the germline, but not in the somatic rearrangement. So that suggests that those events are actually quite rare. The other question we wanted to ask was how good were we at detecting complex structural variants? And just to cut a long story short, when we looked at samples where we had both long reads and short reads, we found that while we did detect a few additional chromothripses and chromoplexies, for the most part, we were actually pretty good with short reads in detecting most classes of complex structural variants. So basically, like I said, three of these statements were really not true. We can actually detect most large structural variants, including complex SVs, and there's very little rearrangement happening between homologous sequences in somatic genomes. So what remains? What is like? As the dust settles, what can we actually do with long reads to understand cancer genome structure? And really, it's this question of phasing nearby rearrangements into long-range somatic haplotypes. And what does that really mean? Well, when we get this graph, it's really an abstraction that summarizes the clonal copy number in a cancer sample. Now, cancer genomes don't actually look like this. They're not graphed structures. This is just the sum of two haplotypes. And in this case, where we have two deletions that are happening nearby in the genome, and this is just a schematic, you can imagine that these junctions actually happen in one of two haplotype orientations. One is that they occur on the same covalently linked molecule, or cis, and the other one would be that they occur in trans, or on two distinct molecules. And each of these reconstructions is equally likely from short reads. What we need are long reads, or in this case, linked reads, which are barcoded short reads that give you the barcodes tell you the identity of the long molecule from which they came, that actually tell us that there are molecules that have both of these junctions in cis. But for that to work, our reads have to be as long as the interjunction distance in order for us to stitch this together and resolve what is a fundamental ambiguity in allelic reconstruction. And so where does this help? One class of variants I identified a few years ago were these clustered rearrangement patterns called rigma, which maybe if you're sort of tempted to call everything chromothripsis, you might say, well, that's a chromothripsis. But there's a few things that that differentiate for this from chromothripsis. One is that these clustered patterns are predominantly involved deletions. They also have a specific biological signature, which they occur in late replicating regions in large multi-megabase genes, and co-occur, but not perfectly, with common fragile sites. So they suggest a distinct mutational process. This is where we can use linked reads, which again is a sort of old technology that 10x gave up once they, jumped on the single cell gravy train, which is probably a good choice. But these give actually beautiful reconstructions of these rigma. And again, we can solve this reverse problem of deconvolving this graph into its constituent haplotypes. And with these linked reads, though we barely make it there, this is the 100 kb scale, we can deconstruct this complex locus into distinct alleles that show that these rearrangements are happening on distinct molecules, suggesting a sort of gradual rather than catastrophic rearrangement process, which is what you would expect with chromothripsis. And here's where optical genome mapping, which takes us to the next order of magnitude of resolving molecule lengths, can actually take the same locus and actually with almost zero ambiguity identify this structure and help us reconstruct this allelic, which actually lets us phase this additional deletion onto this haplotype. And so here, the molecule, these aren't reads, they're actually single molecules that have flowed through a nanochannel where sort of restriction sites have been labeled with a fluorophore, and the interfluorophore distance gives you ability to map to the genome or assemble. And it's a pretty elegant technology that, again, in this very large gene, LERP1B, we can reconstruct this rigma. And so that's consistent with this model of gradual evolution, which we showed in Barrett's esophagus samples from the same patient, which had these rigma that were present in all 4 biopsies in general from each patient, but then contained additional accumulations of new deletions that evolved sort of relative to this ancestral rigma. And of course, what's amazing with Barrett's is that in this pre-malignant condition, you've got very distant, geographically distant lesions with this signature that are clonally related. So Christina Curtis had a beautiful study last year recapitulating these in p53 mutant gastric organoids that these rigma are evolving actually in cell culture, which I think suggests that they are a distinct mutational process that may be what we think is a scar of fragile sites. Now, the definition of a fragile site, I think, has been sort of fuzzy. Everything from anything that showed up on a microarray following ionizing radiation or replication stress in a cell culture model, to what have been called common fragile sites. Now, these rigma don't actually there's not a one-to-one correspondence with this in common fragile sites. So I would argue that these also comprise some rare fragile sites, and seeing even a single example of this in a particular tumor type, I think is sufficient to kind of come to the conclusion that there is a fragile site in that locus. Now, where we apply this notion of phasing, where it's been really useful, is in studying homologous recombination deficiency. There's clinical interest in this because obviously, this is a repair pathway that's essential and that's very important in human cells, particularly in replicating DNA, and tumors through either inherited loss of function in BRCA1 or 2 or acquired can inactivate this pathway and then are susceptible to different DNA damaging agents, platinum chemotherapy, PARP inhibitors, but there's a whole entire new classes of DNA damage response inhibitors that are on clinical trial. And really the question is, what are the backup repair pathways that are keeping these HR deficient tumors alive? And of course, these are breast, ovarian, and prostate cancers, and pancreatic cancers most commonly, but even more broadly, there's been an interest in using this sort of genome-wide profile as a sort of signatures of DNA damage to identify HR deficient tumors. This has been marketed by Myriad in this My Choice test that uses a relatively non-specific signal, which is LOH or allelic imbalance, to identify HR deficient tumors using sort of the genomic profile as a phenotype, right? And that's a sort of interesting concept in and of itself. So this is the only sort of companion diagnostic for PARP inhibitors in specific contexts like in ovarian cancer, but really it's an inferior test to whole genome sequencing, where Serena Nixay-Nall and others have shown that there's actually much more informative signals that we can get from the whole genome. So this HRD index is purely built on these LOH and sort of large state transition patterns. But additional signals that you can get through these sequence level scars are much more informative for diagnosing a tumor as being HR deficient. And I think this is going to be one of the major apps that when we think of sort of the whole genome sequencing as the iPhone or Swiss Army knife of molecular oncology will become useful. Now, the puzzling mystery about HR deficiency is that the fact that HR deficient tumors have a lot of these large scale cytogenetic alterations is actually in line with the intuition from the pre-genome era, where people looked at these cells under the microscope and saw radial chromosomes and all this kind of chromosomal chaos. And sort of the thought is that HR deficiency would give rise to very complex variation, but in fact, the most informative scars of HR deficiency are these very tiny mutations, these deletions with microhomology, which are just small typos in the DNA, or these mysterious signature 3, which occur in a very bland, sort of flat trinucleotide context. Now, there are rearrangement scars associated with HR deficiency, but also these have been very subtle. These are these very simple, non-clustered, non-complex tandem duplications in BRCA1 deficient tumors, and also very local deletions in BRCA2 deficient tumors. So kind of thinking about this paradox of structural genomic instability in cytogenetics and LOH, and then the sort of very subtle HR deficiency scars in seen on whole genomes, we really thought that's sort of a paradox. And we, meaning Simon Powell, who's a radiation oncologist and DNA repair guru at MSK, and Jeremy Seton, who's also a scientist there. So how could we reconcile this paradox in analyzing HR deficient tumors and whole genomes? And we wanted to bring our whole armamentarium of genome graph sort of based annotation of complex structural variants, including some of the new classes, to just compare BRCA1 and 2 mutant tumors to their wild type counterparts, with the hope that we would identify new classes of variants or specific classes of variants that are, especially complex variants, that are enriched in HR deficiency, under this sort of notion that that there would be a lot of complexity. So I'm skipping over a lot of negative results, which I think were interesting, because I think the intuition in the field is that HR deficient genomes would be inherently complex, and we saw chromothripsis in these HR deficient genomes, but we saw it at the same rate in HR proficient genomes as well. We also saw ecDNA. We also saw BFB cycles. So all these variants were present. The only class that we actually saw enriched was this class of variants that we called reciprocal pairs. And there's a few flavors of these, and each had a different predilection for BRCA1 versus 2 deficiency. So these reciprocal duplications are these coupled segments that can exist on a same chromosome or a distant chromosome, where there's this small kind of 10 kb duplication, but then 2 rearrangements that are linking these 2 distant segments. And this is a very BRCA1 deficiency specific pattern, almost never seen in HR proficient tumors. This reciprocal deletion was a BRCA2 deficiency specific pattern, similar to this reciprocal dupe, but now you have 2 deletions and these coupled rearrangements. And finally, this reciprocal deletion duplication, one loss, one gain, that was present in both BRCA1 and 2 deficient tumors and absent in HR proficient tumors. So, one observation about these rearrangements is that they were sort of these quasi-reciprocal rearrangement events. And there's actually quite a bit of ambiguity in how you might reconstruct these in terms of what would be the allelic structure that would give rise to this graph pattern, where again, here for this reciprocal dupe, we have these two coupled duplications on distinct loci. And this is where we thought long reads, in this case linked reads, would help us deconvolve, disambiguate these patterns. So there's basically three possibilities with these loci. One is that you're getting a copy paste event of this E segment in locus 2 in between two duplicated copies of B in this first locus. And to support this, we would expect to see sort of linked reads or long read support showing both of these two junctions in cis, and this kind of stripe pattern on this barcode sharing map that is sort of where each pixel here represents how many long reads or linked read barcodes are shared between pairs of locations in these two loci. So you might be familiar with this kind of plot from high C, very similar concept here for long reads. So this stripe pattern could also go in the opposite direction, which would imply a copy and paste from locus 1 into locus 2. And both of these would be very local kind of copy paste rearrangements, essentially like a duplication. Now, what would be a more profound structural alteration would be this trans pattern, where these two junctions, instead of being on the same haplotype, would now be on distinct haplotypes. And again, we could map this with long reads, and on the barcode sharing map, we would see this kind of colliding diamond pattern. And so, each of sort of eyes wide open, we thought each of these was possible, but we kind of figured that this cis arrangement would be really the predominant. And while indeed we did see this quite frequently, we actually saw this trans pattern around half the time. And so here's some real data from a BRCA2 deficient tumor. We had sequenced about 100, 50 cases, 50 HR deficient cases. And this colliding diamond pattern, you can see with clearly the long read support. Every row here represents a different linked read barcode, with the reads aligning to the various segments, and this sort of gap indicating the presence of a junction, and these junctions are clearly in trans. So this trans pattern was, like I said, occurred about half as frequently as the cis pattern, but actually quite frequently. The other interesting clue was when we did the end-to-end reconstruction of these events, we saw that the cis patterns had a very distinct size distribution, which is that the segment that was copy and pasted in between the two duplicated segments tended to be in the sort of under 1 kb range, while the trans patterns tended to be in the have both rearranged segments in the 1 to 100 kb range. So sort of distinct relationship between phase here and size. Now, what's the meaning of this? Well, I think it's actually the sort of implication of these trans rearrangements is that we may have found a class of alterations that's sort of similar to a reciprocal translocation or reciprocal inversion, with where both derivative chromosomes have duplicated segments from the originating loci, and that's sort of an exotic outcome. And so one thing we wanted to ask was whether this trans configuration was actually creating changes in large-scale genome structure. And remember, our reads here, they're long reads, but they're still at the 10 kb scale. If you zoom out to a chromosome scale, these are just still tiny scars that would not be visible, on fish. So how could we study the long-range chromosomal consequences of these rearrangements? And to me, this is the coolest result from the study. We wanted to ask whether the phase of these rearrangements sort of was correlated with orientation to the centromere. And the prediction here is that these copy-paste events, which are essentially like very local tandem duplications, the orientation of these junctions would be sort of wouldn't care about the location of the centromere. But in the trans case, you can have one of two very different outcomes. One, which would create only monocentric derivatives, and the other one which would create at least one acentric derivative and one dicentric derivative, and this could happen intrachromosomally as well. And so in this type 2 outcome, we would never expect to see a reciprocal, or we would rarely expect to see a reciprocal pair, because one of these junctions would tend to be lost. Of course, there's ecDNA that can be positively selected, but for the most part, this wouldn't really create a selection pressure on average, right? So we would expect to see a depletion of these type 2 orientations in the trans, but not the cis orientation. And this was sort of a bit of a leap, but the data perfectly recapitulated this this sort of intuition. And again, here you can see the enrichment of this monocentric outcome or the depletion of this acentric outcome in the trans, but not in the cis analysis. And this is in our long read data, but actually we could impute phase. I'm not going to show that in our short read data, and we saw a very similar pattern. So what does this mean for HR deficiency? Well, I actually think this is where the phase gives us some insight into mechanism, and this is a bit of speculation. But I think it's a sort of working backwards from the fact that we found a set of very sort of similar topologically related rearrangement patterns that that in BRCA1 and BRCA2 deficiency, and I'm not showing some data how sort of the similar distribution of size with reference to these BRCA1 deficiency associated tandem duplications. But this sort of spectrum of related outcomes suggested that they may be tied to a shared intermediate and a shared sort of mechanism. And what would be that shared intermediate? Well, one of the major roles of HR is in restarting replication forks in all of our normal cells, and these kind of broken forks happen quite regularly because of nicks or DNA adducts. And really, this is a problem that HR deficient cells need to solve, which is this problem of how to deal with a single-ended double-stranded break and how to sort of restart a broken replication fork. And Ralph Scully in 2017 showed that BRCA1 deficiency associated tandem duplications are essentially a response to HR deficient cells trying to restart their fork, restart a broken fork, and this duplication happens because you get a sort of a restart upstream of that original blocked fork, and that aberrant replication restart is what creates this tandem duplication. So what I want to show you is a small variation on this theme could explain the full spectrum of cis and trans patterns that we saw in our samples. And so how do we get to the cis reciprocal duplication? Well, what you can imagine is this single-ended break, instead of reinvading that original collapsed fork, could sort of take a joyride to some nearby, in 3D proximity locus, and undergo a process called MMBIR, where it invades this distal strand and sort of extends for a little bit before undergoing a process that's well characterized in MMBIR, which is template switching. And in this case, again, going back to this original broken replication fork and duplicating this B segment. And that's what would give us this kind of short tract of this distal E segment. Now, another phenomena that's associated with MMBIR is something called half crossover. And this can happen when following this invasion, what this sort of tract of DNA synthesis can mature into a full-on replication fork, and there's several sort of proteins that can be involved in that sort of maturation, like DNA primase and PIF1. So the outcome of this would be that now you've got a full-on replication fork happening at this distal site, and that at some point will collide with whatever is an oncoming, remember, an S phase. So there are lots of 10 to 100 kb sized replication bubbles. asynchronously sort of replicating each chromosome. And there will be a choice. One choice would be that uh, uh, this invading fork gets dislodged and again comes back and gives you a cis outcome. The other would be that actually this distal locus now gets turned into a new single-ended break, which goes back and reinvades this original replication fork. And that's sort of what would give us this trans pattern and sort of extrapolation of the work of Anna Malkova and colleagues. But I think the first association of this with HR deficiency, and of course leading to this phenomena of aneuploidy losses and gains. So we think that these reciprocal duplications are sort of the missing link between the scars, sequence level scars that we can detect with whole genome sequencing that are of course relevant for clinical diagnosis, and this kind of structural genomic chromosomal instability that's been long seen under the microscope. I just want to touch on really zooming out a little bit on larger scale chromosomal structure. And really, we got lucky with this HR deficiency pattern because, you know, the junctions that the phenomena that we were detecting involved junctions that were within 10 to 100 kb of each other. And as a result, we could unambiguously phase nearby junctions into long range haplotypes. But in many cases, we aren't so lucky. And a lot of the most interesting patterns happen on a much larger multi-megabase scale, where I would argue even optical mapping is insufficiently long to resolve that variation. Of course, there's been a lot of interest recently in extrachromosomal DNA, both as a way of amplifying genomes, but also elegant work suggesting that these could be, in many cases, mobile enhancers and have distinct roles in biology. We're actually quite interested in these chromosomally integrated amplicons, and these are just, you know, like BFB cycles. These are different ways of arriving at the same genomic result, which is amplifying an oncogene. A few years ago, we identified another phenomena that we think is distinct from ecDNA and breakage fusion bridge cycles, you know, which are extrachromosomal versus chromosomally integrated mechanisms of amplifying genomes. These Tifonus are massive. They span tens to hundreds of megabases. They have a lot of these fold-back inversions in purple, but they have a lot of high copy junctions, and the junction copy number has this unique pattern where it's almost uniformly distributed from the lowest to the highest junction copy number. And we think that sort of refers to a unique mutational process that's generating these alterations. These have a distinct specificity for specific cancer types, including liposarcoma, but also acral melanoma, which is an interesting tumor type that has a low TMB, but sometimes, not that infrequently, has responses to checkpoint therapy. It's not unknown what's the antigen. One theory could be that these amplified passenger fusions, of which there are many in Tifonus, could be a sort of source of neoantigens in these cancer types. And what's remarkable about these Tifonus is when we've looked at them under the microscope, there are actually no ecDNA. All these copies are concentrated on the end of, in this case, two distinct chromosomes that maybe underwent some duplication following the emergence of this Tifonus, in this case, co-amplifying two of these transcription factors in the small cell lung cancer cell line. Now, we did optical mapping here, but in fact, we could only really guess at the structure. And see, I had an animation here, but oh yeah, there we go. And so sort of predicting that it's sort of a weaving between these two amplified oncogenes, but still there's a lot of ambiguity here, and particularly in this question of, is this locus chromosomally integrated, or is it actually extrachromosomal? Could we actually detect that and prove it from looking at the whole genome? And really, high C was the only way we could unambiguously show that this locus, or at least strongly suggest that this locus was chromosomally integrated. And that's because we took all of the 3D contacts that were originating in the Tifonus amplicon and then looked at where else in the genome that wasn't highly amplified were those 3D contacts going to. And we found a very focal area on chromosome 1q which is indeed the site of chromosomal integration. And you could see this sort of in the off-diagonal contact patterns emanating from the two foci of this Tifonus onto the sort of location of chromosome 1q, where we also see some junctions. And so we've been really wanting to generalize this process, and particularly of working backwards from and forwards between Hi-C and our genome graph models, and really to get at this multi-megabase scale of rearrangements, not just to think about how these loci evolve, but really to try to understand what is their functional role and what could be the sort of positive selection forces that are being applied to these loci. And really, this goes back to that notion of junction copy number. And here I'm showing linked reads, but Hi-C has a similar property, which is that, we infer junction copy number from just looking at these segments, but really the sort of off-diagonal intensity of these sort of contact maps will be correlated. It tells us basically junction copy number tells us where and how much of this off-diagonal pixel density we can expect. And this is where Iraj Eshki, a postdoc in the lab, has recently been developing very cool ways of modeling the folding of these haplotypes that we're inferring from these genome graphs, and then sort of simulating them or predicting them in the coordinates of the rearranged genomes, and then lifting them back to the reference. And this actually gives us, even naively, a surprisingly good map reconstruction of these predicted 3D contact maps. And this is for a very complex locus at the multi-megabase scale. Of course, we still have to model in compartments, and the question is how low do we want to go? But I think this is a very promising direction. And really the next step is the simple question of distinguishing between integrated and extrachromosomal amplicons. I do think that distinction has a sort of impact on how we interpret whole genomes. And I think Hi-C might be the best tool for this, but I think we still need algorithms to do this reliably. But the broader question of, can we reconstruct complex alleles on the multi-megabase scale? I hear ONT might be coming up with 100 megabase, and I don't know, I can't make predictions, but I mean, can we either wait for reads that are much, much longer, or can we use the sort of property of Hi-C where you have a very slow sort of decay of contacts as a function of genomic distance? And then more broadly asking, what are the allele-specific sort of functional shifts in A and B compartment triggered by rearrangement and amplification? I think it's a fundamental question. And are these structural variants evolving to create convergent 3D states? And we've got some technology to look at high order interactions. And there's a great center at the New York Genome Center funded by the Macmillan Foundation, and we're working with Gamze Gersoy on some exciting work. I'm not going to get into POR-C and this, but I just want to finish so we have time for questions. So mass balance, I think, is really the best way to analyze copy number. Even if you're not interested in complex SVs, even if you don't care about a lot of the downstream applications of analyzing these graphs, I think it's just a great copy number caller. And I think, hope people can start using it. I think it's a great way to analyze these whole genomes. Long reads don't detect new cancer genome junctions, large-scale rearrangements, really. And again, our cutoff here is 10 kb. So under 10 kb, yeah, sure. There's a lot of stuff happening with long reads, but for large-scale structural variation, really the value add is to help us phase large structural variants. Here we use this to resolve a paradox in HR deficiency, and use this to kind of work backwards to a potential mechanism that might be a way that these cells survive and is a backup repair mechanism that potentially can be targeted. But long reads also in case of phasing are not long enough to phase the really large variants. I think Hi-C will be more useful here, and in this interplay of 3D and 1D structure, I think genome graphs will play a central role. I want to thank the folks who helped with the data. So many of them have graduated from the lab, but here's the new crew, and we're at 2 locations in New York, and I'm building a new cancer genetics program at NYU. So We're recruiting faculty, so please spread the word, and I'm happy to take questions. Thanks.
Thanks a lot for this fantastic lecture, very inspiring. I see some questions in the Q&A, and please, if you have questions, use either the chat or the Q&A to type your questions. So the first one from Gabriel Funigana. Did you observe similar patterns of reciprocal pairs in non-BRCA altered HRD cases?
Did we observe similar patterns in non-BRCA altered? Yes, we did. So we looked at the long tail of pathogenic alterations in other homologous recombination pathway members, and of course PALB2, the different RAD51 isoforms, and we did see a substantial enrichment, particularly in the biallelic case. So we think that this is a way not only to sort of more confidently identify HR deficiency driven by those other alleles, but also sort of can help us diagnose VUSs, and also determine which of these are more BRCA1-like versus BRCA2-like, which may or may not be a useful axis clinically to analyze these, but I think is something that we can do now with more precision.
Another question in the Q&A. So do you think the different classes of reciprocal pairs will induce different gradations of micronuclei formation?
So could reciprocal pairs be related to micronucleus formation?
To different gradations of micronuclei formation, yeah.
Gradations. So not sure I fully understand the question, but I think there's certainly a way to get to micronuclei with reciprocal pairs, for example, that create a dicentric chromosome, which is an outcome that was depleted in our sort of sequence genomes, but could create that. The only, the interesting negative result in this analysis is we didn't see more chromothripsis, or Tifonus, or any of these really dramatic catastrophic patterns in HR deficient tumors. We saw them also in HR. Now, they may happen through different mechanisms, but as an outcome, we didn't see a higher rate of those, which I think is an interesting negative result. So I don't know if I answered the question there, but just some tidbits, yeah.
I see one more question. Why do you think BRCA1 deficiency or BRCA2 deficiency are not associated with larger complex rearrangements like ecDNAs or chromothripsis?
Yeah, so I think, part of this is there's maybe surprising stability, and, what we're unearthing in HR deficiency, one thing I didn't show is we have signatures of SSA, very strong signatures of single-strand annealing in BRCA2 deficiency, but there are other backup repair mechanisms that maintain genome stability. And, maybe on a single cell level, you're still seeing quite a bit of chaos, but the samples that actually, or the cells that actually are selected out are the ones that are able to, put these additional backup repair mechanisms into place. So I think it's an interesting puzzle. It would be great to get a handle, on how to knock out these additional and mechanisms, see if cells survive, or then we see new variation patterns. But I think the stability is sort of surprising, and also the simplicity of these reciprocal pairs. You can imagine that single-ended break being a live wire that now triggers a chain reaction, but somehow it's relatively contained. We don't see higher order chains in these BRCA deficient genomes. They're generally just pairs.
Thanks. One last question from Jeff. So many of the interpretations of your data start with a biologically motivated hypothesis on what the data should look like. In practice, is this always the case? Or do the patterns in the data sometimes help you define the hypothesis?
I think that's the holy grail, and maybe we just plug the AI, like Roy Kashoni came to NYU recently, and he has a software called Data to Paper, which would be amazing. And then we can just, hang out and drink coffee and stuff. But I think, I do think it would be interesting to have a more algorithmic process to think about mechanism. I do think phase is the intermediate kind of layer. What's the allelic phase, and jumping from that to mechanism, I still think would be tricky, but I think it's an interesting problem. The other question is, how do we characterize recurrent patterns in these graphs and find them de novo? A lot of that we do using something, in my lab I call NI, which is like AI but natural, meaning we're staring and then coming up with criteria. And maybe that sounds kind of hokey, but in a way that's the best we can do. Could we automate that process using like graph neural networks or other fancy machinery from the last 5 to 10 years? I think that's a super exciting problem. One question is, do we have enough data? I would love to have 1 million cancer genomes. I always have FOMO about the GWAS people and their UK biobanks and all these things. When can we get to those numbers? We might need to get to those numbers in order to apply these kind of fancy tools.
Thanks a lot. I don't see any more questions in the chat, so I'd like to thank you again for this really inspiring lecture, and thank you everyone for joining. And the next talk will be on the 19th of July. We'll send the link around. Thanks a lot, and have a nice rest of the day.
Thank you, everybody. Thanks.
Thank you, Marcin. Thank you so much.
Thanks. My pleasure.